Thank you for the invitation to speak today, and I'm looking forward to the interactive session this afternoon. What I'm going to tell you about are technologies that we think are important for the ability to integrate many different kinds of data, not just omic data, but also clinical data, to be able to understand diseases and develop new therapeutic strategies. And I'll speak largely in the context of neurodegenerative diseases, although the approaches are broader than that. And I want to sketch a spectrum of neurodegenerative diseases. On the one hand, we have somewhat mysterious diseases, shown here as ALS, amyotrophic lateral sclerosis. And these are, at this end of the spectrum, diseases where we really understand very little about the causes. So ALS, like many other neurodegenerative diseases, affects both people who have a family history of the disease, but the vast majority of the patients have no family history. And in ALS, there are 30 genes that can cause, individually, can each cause the familial form of the disease. And in the rest of the cases, what are called sporadic cases, we find those genes with some low frequency. But about 85% of the patients, there's no genetic reason that we are able to identify that the patients come down with the disease. And it's a devastating disease because most patients survive for only about 3 years. Some patients, though, like Stephen Hawking's, some of you may be familiar with, survive with it for decades. And understanding that diversity of the disease itself is also an important part of the problem that we want to be able to study. So if you have a disease with no known cause, it's obviously very difficult to build experimental models or develop therapeutic strategies. At the other end of the spectrum are diseases like Huntington's disease, which are actually quite well understood in terms of their genetics. So the gene that causes Huntington's disease was the first disease-causing gene to be mapped to position in the human chromosome. And that was done almost 30 years ago. And in all that time, we've developed, the field has developed a deep understanding of the molecular and cellular biology of Huntington's disease, but we actually still do not have a treatment that can stop or reverse Huntington's disease pathology. And both of these diseases, ALS and Huntington's, invariably lead to death. So if the conventional approaches to understanding these diseases have been quite challenging and have led to somewhat limited results, I think the hope that brings this all together is the idea that this multi-omic approach could do better. So if we have not just genomic data, but we also bring on data about RNA, about epigenomics, about proteins, about small molecules, and information about the interactions of these molecules from protein-DNA interaction assays, protein-protein interaction assays, as well as high-throughput screens, that perhaps we can take all those data together and get insights into a disease. And on the one hand, this has been a very successful strategy in understanding basic biology. So if you think about what's understood today about, say, transcription compared to what was known before these technologies became common, it's revolutionized our understanding. We understand now that transcription is not just about promoters and genes, but about the entire genome. And similarly, the interest in the microbiome has really been advanced dramatically by the fact that it's possible to sequence DNA out of almost any kind of sample. As you get closer and closer to therapeutic impact, we know that multi-omics approaches have played a role in trying to subtype different types of cancer. And that has a very important impact on our understanding of the disease. But so far, there's really been quite little effect on being able to actually design therapeutic strategies from these technologies. And our goal, and I think the goal of many in this meeting, is to be able to identify those therapeutic targets by an integration of many different types of omic data and do a better job than the field has been able to do so far in finding therapeutic strategies for really, really challenging diseases like ALS and Huntington's. So in today's lecture, what I'm going to tell you about are some of the challenges that we face trying to bring together these types of data. And then one of the strategies that my lab has been pursuing over the years, which is using networks to integrate the data. And I'll show you how we've applied that in those two diseases I've already mentioned, Huntington's and ALS. What are the standard approaches for dealing with multi-omic data? Well, I think one approach is obviously to look for correlations in the data. Any of you who have ever used a heat map have obviously used correlations to find sets of genes or proteins that are co-regulated, and also to find samples or conditions that seem to be responding in a similar way. And that's a very powerful technology. But as we think about therapeutic interventions, we know that correlation is not enough. We need to actually understand causation in order to figure out what the best targets are. Another very important approach to high throughput data sets is to try to interpret those in terms of known pathways. And there are quite a few databases that are quite helpful in this regard. And some of the ones are listed down below: Gene Ontology, MSigDB, and there are commercial databases as well. So what's the limitation of this kind of approach? Well, as I'll show you in a moment, the databases, even the best ones, capture really a tiny fraction of the true biology. And one example that shows that is a paper that we published a little while ago, where we looked at an extremely well-studied signal transduction pathway. This is the EGF receptor. It's a tyrosine kinase. It gets activated by being bound by EGF. And when the tyrosine kinase is turned on, then there'll be phosphorylation throughout the cell on tyrosines. And so we asked, when you actually do the experiment and measure phosphorylation on tyrosines, what fraction of those events that are clearly responding to EGF, the ligand we just added, are in the pathways that are annotated as being EGF responsive pathways? So what fraction of the phosphorylation events are on pathway and what fraction are off? And regardless of which databases we use to make these assignments, we found that typically only about 15% of the phosphorylation events were on the annotated pathway, with 85% of it being off the pathway. So what's the implication? It means that when you take your data and plug it into these databases, some of the information you get will presumably be correct, but a lot of it will just be unannotated. In fact, we find that a lot of the phosphorylation events that occurred in response to this very, very well characterized signal were not annotated in any pathway whatsoever. So it's not even that you're going to get necessarily the wrong answer, you just won't get any answer. So there's a lot of unexplored territory remaining in biology as we try to understand this. So we don't want to just rely on correlations. We can't rely just on databases. Another thing that people often do when they're dealing with multi-omic data is they try to zoom in on a subset of the molecules that they think are going to be the most interesting because they're overwhelmed with the many different kinds of data that they're dealing with. But a study that we did a number of years ago was that looking at different kinds of omic data showed that the overlap between different data sets often is quite a lot less than you would expect even by chance. And this was work that was done by S. Dieger-Lotham and Laurivo when they were postdocs in my lab a number of years ago. And what they showed was when they were looking at genetic screens on the one hand for genes that were essential in response to some perturbation, and the genes that changed in gene expression in response to the same perturbation, there was less overlap than would be expected by chance. And they were able to trace down why that was by looking across a very large number of perturbations. And they were able to show that different assays actually were asking completely different questions about the biology. And so in a genetic screen, you're often asking, what are the master regulators for response? And when those master regulators are knocked out, you don't get the response. Whereas, say, gene expression is often looking at effector molecules, not the master regulators. Many of these are redundant, and when you knock them out individually, you don't see a phenotype. So what do you do when you're faced with these different kinds of data? Well, one of the things that SD and Lauer started in my lab was the idea that maybe we could look for connections among disparate types of data through a network approach. And the idea would be, if we have some cartoon of the cell here where there are some signals that are detected, say, by proteomics in one color and by metabolomics or RNA-seq in another color, there might be very, very few molecules that are detected by more than one assay, which is why no molecule here has multiple colors. But there might be pathways that really do connect all of the biology, where some of the response is seen, say, in the proteomic assay, and other parts of the response are seen in the gene expression assay. And so our goal is to try to recover these pathways de novo without reference to a database of annotated pathways, because those are incomplete. And if we do a good job of this, then along the way, we'll pick up a lot of other molecules which were invisible to the assays that we had chosen, but might still be very important parts of the response. So that's the motivation of the strategy that we take. And the way we do that is we use an interactome. And this interactome in our use is built of physical associations of molecules. And the reason we like physical associations is because they get us closer and closer to causality, which is obviously what we need for the therapeutic insight. And at the same time, though, these interactions are much less biased than the annotation databases that I referred to earlier. And so these come from high throughput experiments, for example, affinity capture mass spec, yeast-2 hybrid, or perhaps even computational predictions, such as a kinase and a substrate interaction. And when you gather enough of these, you find that there are hundreds of thousands, or depending on how you do it, perhaps even 1 million interactions that you can put together in these databases. So these physical associations give us a scaffold on which to try to interpret the multi-omic data. And I'll show you a lot of networks as we go forward. So in these networks, obviously the nodes, the circles are going to represent the molecules. So there could be proteins, they could be metabolites, and then the edges will always represent these physical associations. Now, one critical decision we have to make is what to do with gene expression data. And for a while, the field used to like to map the gene expression data onto the proteins. But as we gather more and more data, we realize that gene expression is a very, very noisy proxy for the protein levels. And as we are able to collect more and more proteomic data, there's less and less need to map the RNA onto the protein and use it as that noisy assay. And so we focus on using the RNA data as a proxy for transcription to try to understand what genes are being turned on and off, what the master regulators are that are turning those genes on or off, I should say. And so we don't map the RNA onto the protein part of the network, but instead we try to reconstruct what those transcriptional regulators were by a combination of gene expression data, epigenomic data, and sequence analysis. And then once we figured out what the transcription factors are that were active in a particular assay, then we can map that back onto this interactome and look for protein-protein or protein small molecule interactions that connect the transcription factors to molecules that we may have detected in the mass spec or some other approach. So if that's the strategy, how well does this actually work? So I'll show you some figures from a very early paper we did in this field where we had a couple of dozen molecules that were changing in a tumor. We had an interactome, and he asked, could we connect the molecules that we were detecting through the network? And the answer was both good news and bad news. The good news was that it was definitely possible to connect all of them. The bad news was that if you just do something simple like a shortest path approach, you get an enormous network out the other end. And so you've taken many tens of things that you didn't understand very well and turned them into many hundreds or thousands of things that you didn't understand very well, which is not a great strategy. So why do we get these really complicated networks that sometimes we call hairballs? Well, obviously, part of it is because the omic data that we're starting with has some false positives in it. But that's actually a small part of the problem. The real part of the problem actually comes from both false positives and false negatives in the interactome. So a false positive in the interactome means that there's an edge there that doesn't really exist, and that obviously is going to connect things that shouldn't be connected. A false negative in the interactome is actually in many ways a bigger problem. Because if you have two molecules that really do interact with a tight connection, but you're missing that interaction in your network, and you force your algorithm to find some way to connect them, it will find a connection, but it could be a very, very remote connection. And so when we demand that the algorithm connect up everything, we basically force it to come up with very implausible, unlikely interaction connections. So if we think about this and zoom into some little tiny piece of that interactome, maybe it'll look like this, where the data for which we actually, the proteins for which we actually have data, say from mass spec, are shown in yellow. There are a lot of proteins for which we have no data in gray. And maybe the true interaction edges are the ones in purple. And there are a lot of other edges there as well. And there's this molecule on the far right of your screen, which may or may not really be part of the same network, but we detect it in the assay. And if you force the algorithm to connect everything, obviously it will. But what if those edges are not very reliable that connect it? Or what if the signal itself is pretty weak? Well, connecting is not necessarily the right strategy. So the approach that we came up with is something called a prize collecting Steiner tree or Steiner forest approach. And this comes from a different, from algorithms used in the telecommunications industry, actually. The equation that we're trying to optimize is shown on the bottom of the screen. It's a simple equation. The idea is that we assign a prize for every node for which we have direct data. So those are, say, the yellow nodes. And if the algorithm manages to connect that node to the network, it gets to keep the prize. But it has to pay for every edge that it's using. And if the edge that it uses to bring that in is unreliable, then it's going to be a high cost for the edge. If the edge is reliable, then the edge is cheap. And so in this case, then, the algorithm can weigh whether it's worth it to try to capture the prize by including that node on the far right. If the edges are high confidence, then the edge costs are going to be cheap and it's going to be worthwhile. If the edges are unreliable, then the edge costs are going to be expensive and it's not going to be worthwhile to try to collect that prize. So this is the optimization that the algorithm does. And we layered on top of this many kinds of statistical tests, robustness, to make sure that we're not finding just one network that might be arbitrary, but we're trying to search over a space of networks as well. So how does this actually work? Well, in that example that I showed you before, where we took a couple of tens of things and ended up with this enormous network of thousands, we got a much smaller network, and that network had an obvious biological interpretation. Now, in those networks, we have a couple of different kinds of things. We have the nodes that were actually measured experimentally. Here they're filled in with yellow, they're outlined in red. Those are what we'll call terminals. They're the ones that have the prizes. And then there are nodes that the network adds on because it's needed to make the connections. And those are predictions. Sometimes I'll call those Steiner nodes because of the Steiner tree algorithm. So this gives us both ways to understand the connectivity among the signals that we received and also some predictions that we can use going forward to try to perhaps get a deeper understanding of the disease. We've packaged this software up over the years into an open source package that we call Omics Integrator. And one of the things I think is an advantage of this method over some other network kinds of approaches is that it's first not relying on correlations. That's obviously very important. So it's mechanistic. It's quite flexible. We can add other kinds of information into these networks, into the optimization. We can even incorporate negative data. So if you know that some protein is definitely not part of the network, that can be embedded in the optimization. Or if you think it's unlikely, you can have a soft penalty as well. And it's very useful for small numbers of patients where you can't do some massive correlation even if you wanted to. So I encourage you to take a look at how this can be applied perhaps to your own data sets. One thing that we got interested in was the power of untargeted metabolomics to give us another insight into what's going on in cells besides some of the more obvious assays that are popular, such as RNA, epigenomics, and proteins. And the importance of untargeted metabolomics is that it's an active measure of many processes, of the current activity of those, and not just the potential activity that might be, say, a protein that hasn't yet been turned on or a gene that hasn't yet been expressed. So if we think about how to incorporate metabolites into our network, well, they're just molecules like any other, so they can be represented by nodes. And if we have protein metabolite interactions, say knowledge of enzymes and substrates, then we get edges from there as well. So that's metabolites, but in untargeted metabolomics, we're using mass spec to detect the metabolites. And most of those metabolites, then we get a mass, but we don't necessarily know what they are. And so how can we incorporate these unknown metabolites in our network? And so there was a student in my lab, Leila Pirhaji, who now is CEO of a startup company that we founded. And the idea that we developed together was the idea that, well, we can add to the network a node that represents not the metabolite, but the actual experimental data. The experimental data is a mass spec peak that tells me a mass to charge ratio. And so I'm going to add one of these triangles to my network that represents the signal that this mass charge ratio is changing. And I'm going to draw an edge between that signal and every metabolite that is a good match based on the mass. Now you'll see in this toy example then that there are some metabolites that are off to the right of the screen that are not connected to any other signals. So if you have other omics data, you can start to distinguish between the likely mass to charge ratios that are matches and the ones that are less likely. So the connectivity helps you try to figure out that some of these assignments are more likely than others. The other thing you can do is apply some of the network approaches that we've developed to look at how robust different parts of the network are. And it may turn out then that by doing some of these statistical tests, where we say add noise to the network, we do randomizations, we may find that certain parts of the network are more robust across different parameter settings. And so we may get increased likelihood that one of these assignments is correct and another one less likely. So Leila tested this out in the context of Huntington's disease, which I already mentioned. There's a gene that causes it. That gene has a trinucleotide repeat, CAG, that encodes glutamine. And all of us have a repeat of that CAG. People who develop disease have much longer repeats than people who don't develop the disease. We used a cell culture model of the disease where we have cells that either have 7 of these repeats, so it's called 7q for 7 glutamines, or 111q. So we used this cell culture model, and we collaborated with quite a few people that were members of my lab who did many of these experiments, and a lot of our Huntington's work is done in collaboration with David Hausman and Leslie Thompson. But Leila was really the pioneer here. And so we collected phosphoproteomic data in this cell culture model. We collected information about lipids that were changing this model. And we asked whether the network models we get out give us any insight into the disease. And so we can separately make proteomic networks, lipidomic networks, and combine them. And as you might imagine from what I told you earlier, that the combination of these data sets is a much richer view of what's going on in the cell than the two separate data sets. So separately, proteomics and lipidomics seem to tell us completely different stories, much like the things that Esty and Laura had found in the lab looking at very different kinds of data a long time ago. But when we merge the two data sets together, we do see a lot of interesting overlap. And we capture most of what we would have picked up with the separate data types, but we pick up more as well. So here's a piece of the resulting network. The pink triangles are the ones that represent the masses from the mass spec, and we have edges to molecules that the algorithms thinks are good matches. And the size represents the robustness, so our confidence in the assignment. And we went back with the different kinds of mass spec technology that allowed us to specifically look at these particular molecules, and we were able to confirm in each of the cases that we tested that the lipids were in fact changing statistically significantly between samples from the HD and the control. So those are molecules that were directly measured by the mass spec, and our algorithm helped us figure out the assignment. But there were also molecules, I referred to them previously as Steiner nodes, such as the one that's outlined in green and towards the bottom, where there was no direct experimental measurement, but there's a prediction made by the algorithm that it's important in the process. And we looked at this one in particular, the sphingosine 1-phosphate, because there's a biology surrounding this that's known in other neurodegenerative diseases. And so we asked whether, in fact, it was changing. And sure enough, although it was tough to experimentally measure, we were able to find a statistically significant difference between the HD and the control samples. And this was particularly intriguing because there's this drug that's used to treat MS that targets this pathway. And so we carried out a number of experiments, both in our own lab and with collaborators, to evaluate to what extent does targeting this pathway have an impact on the toxicity of the Huntington gene. And we were able to show both in cell culture models and a rat brain slice model that it actually does ameliorate the toxicity of Huntington's protein. We, in fact, had another study that we were engaged in analyzing completely different kind of data in Huntington's disease, which also pointed to this pathway. And so we have increased confidence that this could be an important therapeutic pathway for Huntington's disease. So that's in the context of Huntington's. I'd like to tell you now about how we've been applying similar approaches and advancing them in the study of ALS. And this is collaborative work with a number of different groups around the country. It was originally funded by an NIH grant called Neuralinks, and now it's grown into a much larger effort, something called Answer ALS. As I've told you, ALS is a disease in which neurons die. They're, in this case, the motor neurons, the ones that go from the brain, the upper motor neurons, through the spinal cord or the lower motor neurons and control the muscles. And in patients, invariably, you have the failure of these motor neurons ultimately leads to the inability to even breathe. To understand the disease, we'd like to be able to sample motor neurons and measure by all these omics technologies what's going wrong in these cells. Now, what kind of neurons would be the most interesting? The ones that would be most interesting would be the ones just on the cusp of neurodegeneration, right? Once the disease has progressed, the neurons are dying, there may be nothing that we can do to stop it. But these diseases tend to occur, ALS in particular, tends to occur in middle age or later. And so there's the potential then if we could figure out that the disease was starting, perhaps we could block degeneration. So we want to look at early stage neurons, but we can't get at them. We can't biopsy people's spinal cords to measure these things. And so the technology we use is induced pluripotent stem cells. And in this technology, then we take cells from a patient, we make them into pluripotent cells in the lab, and then force them to differentiate into motor neurons. And then we measure many different kinds of omics data. So we have gene expression data from RNA-seq, epigenomic data from ATAC-seq, and then proteomic data from mass spec. And now we can try to ask what are the pathways that are altered in the early phase of this disease. And we focused on one particular type of ALS. I told you that there are a number of genes that can cause the disease. The most common disease-causing gene that's known so far is something called C9orf72. And so we looked at cells from patients who have that mutation and also at controls. In the lab, our collaborators differentiated these into motor neurons. And then in Leslie Thompson's lab, they did RNA-seq. In my lab, in Jenny Van Eyck's lab, they did proteomics. In my lab, we did ATAC-seq. And then Steve Finkbeiner's group is doing single cell live imaging on these same neurons. So what can we learn from these data? Well, we get a lot of heat maps. That by itself has only limited potential to give us insights. But a graduate student in my lab, Johnny Lee, took charge of making sense of all these data. And so he used the Omics Integrator strategy. He adapted in some ways, did a lot of tests for robustness and significance, and tried to understand what the networks were telling us out the other end. So we're going to take the RNA-seq data, and we're going to try to identify the transcriptional regulators that are turning those genes on and off by combining that with the epigenomic data from ATAC-seq. And so when Johnny did that and merged these data sets, he found a number of transcription factor motifs. And then he asked, what do we know about the proteins that might bind these transcription factors? And do those proteins, the transcription factors themselves, change between the ALS and the control samples? What we found was really quite striking. So in this plot, on the x-axis is the fold change in the RNA for the transcription factor. And on the y-axis is the statistical significance for its motif. And you can see that there are a lot of transcription factors that are more highly expressed in the ALS samples, and their motifs tend to be in regions that have more open chromatin in the ALS samples. And conversely, there are ones that are more expressed in control samples, and their motifs are in regions that have more open chromatin in the control samples. So with this model of transcriptional regulation, we can then try to connect it up with the proteomic data that were detected in the assay. And so we put that into the omics integrator pathway. We end up with networks. And how do we decide whether a network is actually telling us something meaningful? So Johnny's idea was to gather as much information as he could from the literature, from genetic screens that were done in model organisms, from a database called Open Targets, gathers information from the literature, and see whether the proteins, the molecules that we were picking up in our networks were likely to be part of ALS-related biology or not. And what he found were our networks were very strongly enriched for molecules that had been previously shown to be associated with ALS biology. And you can see the p-value there. And in this network, the color represents the change in the proteomics or our signal from the predictions of the transcription factors between the ALS and the control. And the borders around the molecules are colored based on whether there's prior genetic data or whether it shows up in the Open Targets database that indicates it is related to ALS. So if we zoom in on one of these pathways, extracellular matrix, here you can see a very, very coherent response in the cells where the ALS samples have much higher levels of expression of these proteins. And a few of these molecules were previously associated with ALS, but not very many. And that's interesting, right, because it means that a lot of what we're discovering is new, which is really great. But we haven't missed important aspects that were previously identified. You can see that in other parts of the pathway, the response is more mixed. There's some proteins going up and some proteins going down, and we'll come back to that later. One aspect of the network that struck us as quite interesting was about actin organization. And one of the things that I mentioned previously is that there are a lot of different proteins that change that can cause ALS. So mutations in almost 30 different genes, any one of which can cause ALS. The samples that these were taken from were patients who have the C9orf72 mutation. But one of the proteins that we detected as changing was this one, TUBA4A, which is a microtubule-associated protein. And this one is actually a cause of ALS in other patients as well. And so we can see that sometimes these genetic causes may be converging on similar pathways, and maybe some of the perturbations that are caused by C9 overlap with some of the perturbations caused by other mutations that lead to ALS. And it wasn't just that one protein. There's actually a whole network of proteins associated with tubules, microtubules, I should say, as well as a lot of the regulatory proteins. So we see microtubule-associated protein that's a regulator that has also been associated with ALS in other studies. We even see on the bottom a master regulator of these regulators of the microtubules. So there seems to be a lot going on in this actin organization network. How do we follow up on this? So we've pursued a number of different strategies, and the one I'll tell you about today was done in collaboration with Tom Lloyd at Johns Hopkins. And Tom Lloyd uses a fly model to try to understand ALS. And we thought this might be actually particularly useful in trying to identify therapeutic insights. And the reason why is in the networks that I've given you, we can say that maybe some molecule is a good point of intervention, but we can't tell you whether you need to knock it down or overexpress it, right? How do we know which ones we should be inhibiting, which ones we should be activating? So we used Tom's system in which he overexpresses the repeat that causes ALS in these C9orf72 variants. It's a 6-nucleotide repeat, G4C2. He overexpresses that in the fly, and it leads to mild degeneration of the retina. And then he knocks down different genes, and he can see whether that knockdown actually makes the phenotype better or worse. So what happens if we take the data from our networks and we look at proteins that we detected as changing, and we ask, when you knock them down in a fly, what happens? So you might imagine, for example, we have a protein that went up in the ALS samples, and if we knock it down, it suppresses toxicity in the fly. Well, that seemed to indicate that it's not a causal pathway, right? It went up in the disease samples, knocking it down makes the fly better, maybe knocking it down in a human would also make the person better. I want to distinguish that from, say, an opposite case where it was up in the ALS samples in people, and knocking it down makes the fly phenotype worse. Well, what might that mean? Well, that might mean that it's a compensatory mechanism in the people, right? So it went up in the people not because it was on the pathway that was causing the disease, but the cell was fighting off the consequences of the disease. And that would mean that potentially knocking it down in the person would be a bad strategy. Maybe you would need to activate it. So we can distinguish between causal causal proteins and compensatory proteins in our networks. So we did was Tom's lab gathered quite a lot of fly data. And then Johnny distinguished between the potentially causal and compensatory proteins in all of these. And then he went back to the networks and developed a network level view of what was going on. And just to summarize that in a cartoon version, what we find is that we can go from the C9orf72 mutation and identify certain pathways that really are on the causal pathway. And some of these match what's been known previously. So nuclear cytoplasmic transport is disrupted in the C9orf72 patients, and our analysis shows that that is part of the cause of the disease. Whereas the stress response proteins are altered, and that's part of the compensatory mechanism of the cell trying to fight off the disease. So we think this is a very important piece of network biology, which is not just to get an answer that says, here's a network, but to really try to tease out the causality. And it was quite important here that we had additional experimental data that helped us to interpret the causal relationships. So with these technologies in hand, we can now try to zoom out to the big picture in ALS. And I told you that this was one mutation that causes ALS out of many. And even altogether, all these mutations account for a tiny fraction of all the people who have ALS. So how many types of ALS are there? There are a lot of these genetic variants. I haven't gone into much detail on it, but there are quite a lot of clinical variants as well. So, on the one hand, we have people like Lou Gehrig, who passed away very, very quickly from the disease, Stephen Hawking, who passed away slowly. There are a lot of clinical variants in terms of where the disease begins in the patients. Does it begin in the arms, the legs, the bulbar region? What processes, is it fine motor, gross motor, or speech that get affected first? And people have looked for a long time at relationships, whether there are relationships between the genetic variants and the clinical variants. And it's been quite hard to find anything. There are a few exceptions, but by and large, a clinician cannot tell what mutation the person has or whether they're a sporadic case. Our hope is, though, that we can identify certain types of pathways here that would distinguish these patients. And why do we think this is important? Because in direct analogy to what's been found in cancer, we think that when you can identify the pathways that are disrupted in a tumor, you can have a better insight in how to treat that tumor. And one treats different types of breast cancer with different modalities. One treats different kinds of lung cancer with different modalities. We hope to get to a point where we can figure out the different types of ALS that there are, and then know the networks that are altered in those different types of ALS, and find therapeutics that will work in those specific subtypes to target those specific pathways. So to do this, we expanded our collaboration beyond the original researchers who were in Neurolinks, and we have a big clinical research operation as well. And together, all these clinicians have brought 1,000 people into their clinics. We've recruited 1,000 patients, I should say, and quite a few controlled patients as well. And what do we do with each of these patients? Well, one thing we do is we generate IPS lines from each patient. And so that's proceeding apace. It was slowed down a little bit by COVID, but we're still working at it quite well. And so there are several hundred IPS lines already available from these patients and from controls. What kind of data do we collect? Well, in addition to taking their cells into the lab, we gather clinical data from these patients. So we learn a lot about their disease progression. We've also started using a phone app to monitor some behavioral symptoms. And so one really exciting aspect of this is trying to measure some cognitive changes by showing the patients a picture, like the picture shown in the middle of your screen, and asking the patients to describe what they see. And the IBM Watson team shown in the lower left have been analyzing those data and can actually identify certain cognitive changes in the patients based on how complex their descriptions are and other features. Then we also collect some standard reading passages to measure their ability to speak. And we have some fine motor skill tests as well. And this is an evolving field, and we're exploring other technologies besides phone apps for this kind of remote monitoring, which I think is going to be increasingly important in understanding the full picture of the disease. So we have the IPS lines that I told you about previously. We gather epigenomic proteomic and transcriptional data from them. We had collaborated with New York Genome Center to get genomic data on all these patients. We gather clinical data through an organization called Neurobank that helps refine those data. And then the phone app gives us behavioral data. And our goal is to find the subtypes of ALS. That's part of the goal. The other goal is to make these data available to anybody. And so if you go to this website, you can see a portal that was built by someone in my lab, Alex Linné, and you're welcome to download the data. You can also, from this portal, order some of the IPS lines and do experiments on them yourself. And there are tools both for finding out what data we have and also some basic visualization tools on the website. Up to now, I've told you largely about how we analyze the molecular data, but we think it's also going to be important to identify clinical data. And one of the things that's critical to understand ALS is who progresses quickly and slowly. The standard measure that's used for that is a rating scale called the ALS-FRS-R. It's a rating scale that's developed by asking patients questions about what things they're able to do on their own. And then you can get a score from that that goes from 0 to 48. And you can look at how that progresses over time. And patients will always decline over time. But we want to understand whether patients are declining rapidly or whether they're declining slowly. Now, that turns out to be harder than it sounds because the actual amount of time that we see these patients in our studies is rather limited. And so we don't have that longitudinal signal over a long period of time, and there's a lot of noise in this kind of subjective assay. And so Divya Ramamurthy in my lab, together with another team from IBM Watson, developed ways using a mixture of Gaussian process model to group these patients together into fast and slow progressors. And we can actually go into an enormous database of clinical data called PROACT and show that there are quite a lot of different patterns of decline that can be seen, most of which are not linear, which is what is typically used to model decline in this disease, but some which have profound non-linearities. I think that's going to be important in understanding progression. And then the other aspect of this is that we want to then try to relate those clinical parameters to molecular parameters. So the Answer ALS program, we've completed the enrollment and the data are available and the data collection is in progress. And now we really think we're on the cusp of figuring out how many different molecular types of ALS there are. That may come from analyzing purely the omic data, but I think it's going to be more likely it's going to come through a tight integration of analyzing the molecular and the behavioral data. And then the next and most important step is if we can identify those subtypes, can we identify the interventions that would halt neuronal death? So just to quickly summarize what I hope I've told you today, we believe that networks play an important role in trying to find complementarity in data sets and going beyond simple annotation-based approaches. And they're quite powerful in interpreting cases where you have small numbers of patients and also interpreting data that historically have been very hard to make sense of, like untargeted metabolomics. But they're not enough. We're not just a one technology lab. We believe that there are a lot of other things that have to come into play. We think that additional experimentation, additional computational approaches are needed to make sense of the causality and to identify the therapeutic strategies. And I've shown you how these approaches have led to some insights into therapeutic potential of a particular pathway in Huntington's disease and the progress that we're making towards identifying therapeutic strategies in ALS and our focus on identifying subtypes of the disease, especially among the sporadic patients where the genomics are not informative at this stage. So with that, I want to close. I want to thank all the people in my group who I haven't had a chance to mention and my great collaborators. And I also want to mention we have postdoc positions available for people, primarily experimentalists, who'd like to get deeper into this interface of experiment and computation to really take that next step to identify the causal pathways and develop therapeutic strategies. And I look forward to speaking with you later this afternoon during the question and answer period.